So Chapter 6 will cover some topics that you're probably familiar with, different aspects of chromosome variation. All right, there's different types of chromosomes, and we've talked about this a little bit in lab, and the chromosomes are classified based on where the centromere is positioned, and we have different types of chromosomes. For example, we call them metacentric if the centromere is positioned in the middle of the chromosome. Submetacentric is where the centromere is slightly displaced to one side so that you have a short arm and a long arm that are identifiable in that way. You have acrocentric chromosomes where the centromere is completely at one end, and you have telocentric chromosomes where the centromere is completely at the end of the chromosome. In humans, we don't have any telocentric chromosomes. We only have metacentric, submetacentric, and acrocentric. But other species have telocentric. And so when you want to look at chromosomes and identify them and classify them and name them, as we do in human genetics, you have to be able to see the chromosomes. You have to stain the chromosomes. And so the way that you stain chromosomes is part of a process we call karyotyping. So in order to do a karyotype, you have to grow cells that you cause to become stuck at metaphase, and at that point the chromosomes are really condensed, and then you drop the cells onto a slide and the cells burst open, and you can fix those chromosomes from the cells onto the slide and put stain on them. And there's different types of stain you can use, and the different types of stain will stick to different parts of the chromosomes, and we call them different types of banding. So the typical type of stain that we usually talk about in beginning genetics is called Gamsa stain. G-banding chromosomes means that you stained it with Gamsa stain. And you get this stripy pattern you can see on the chromosomes here. And you can see the centromeres to some extent. Q-banding chromosomes uses a different stain that's going to stick to different types of structures on the chromosome. C-banding chromosomes means you stain the centromeres. So that's especially useful if you just want to count how many chromosomes are in each cell. And there's something called R-banding. So there's different types of stains you can use that will stain different things, and mostly what we're going to see is G-banding of chromosomes, because that's kind of the classic or one of the earlier stains that was used, but just be aware there's other kinds of stains. But you can stain the chromosomes, and then you can measure them. You can look for these different striping patterns to help you identify or distinguish between the chromosomes, and that's all part of the karyotyping process. But what we're going to talk about in Chapter 6 is variations of what would be considered normal chromosome structures or numbers. And so everything in Chapter 6, we talk first about normal chromosomes, and then we quickly go into unusual chromosome structures or unusual chromosome numbers. And there's really three categories. You can have chromosome rearrangements, so rearrangement within a single chromosome, and the other chromosomes are pretty much unaffected. So that's shown here on the left, chromosome rearrangements. And you have different types of rearrangements here. They're showing what's called a duplication. Then you can have an unusual situation where you have the wrong number of chromosomes, but only for one chromosome. So like here you have the blue chromosome is present in 3 copies, but the other chromosomes are present in 2 copies. So you have a diploid number of chromosomes, except there's one extra. So that's called aneuploidy. If you have an extra chromosome, it's called a trisomy, for example, if you have 3 copies. Or you can have a monosomy if you only have 1 copy. But if you have an extra copy or a missing copy of just a single type of chromosome, that's called an aneuploidy. And then you can have situations that are called polyploidy. Polyploidy is when you have multiple sets, but everything is still in balance across the different types of chromosomes. So if you have 3 copies of the blue, you have 3 copies of the pink, and 3 copies of the green. So 3 sets would be triploid. So polyploidy would be something along that line. So there's different things that can happen at different stages of development and in different cells, and they have different effects. So we're first going to look at the structural abnormalities, the deletions, the duplications, the inversions, translocations, fragile sites, and I didn't add on copy number variants. So a deletion or duplication means within a chromosome. I'm not talking about an extra chromosome, an extra entire chromosome. I'm talking about within a chromosome, you have a section of the chromosome missing or extra. So you see here, if you have a normal chromosome A, B, C, D, E, F, G, then if there's a duplication within that chromosome, you have A, B, C, D, E, F, G. So that one chromosome is affected. But you're not getting another copy of a chromosome, you're just getting one chromosome that is a little longer. So you still have two copies, let's say, of that chromosome, but one of the copies is too long. Or a deletion within a chromosome. Not a deletion of an entire chromosome, but a deletion within a chromosome causes you to get one chromosome that's shorter. So missing a section but not a whole chromosome. Duplicating a section but not duplicating the whole chromosome. So when we talk about chromosome rearrangements, duplications, and deletions, that's really what we mean. Now, usually there's not going to be any problem in meiosis with an individual who has a normal chromosome and then a homologous chromosome that has a duplication or deletion, because there will be the formation of what's called a compensation loop that will allow for meiosis 1 to occur normally, so you'll have normal synapses. So for example, in this case where you have a heterozygous duplication, the chromosome that has the extra section will just loop out, and that will allow the synapses to occur pretty normally. But what's going to happen here is that even though meiosis is normal, you have a problem immediately in that individual. So even though they're fertile, they have a problem because you're upsetting the amount of gene product that's in the cells. If there's too many copies of a certain section of chromosome, that means you have extra copies of any genes in that section. And so you're upsetting the balance within that cell immediately. So that individual is fertile, but they're maybe not normal themselves. So an example of this is called BAR, the BAR eye, which was discovered in Drosophila. And the reason the BAR region of the chromosome is called that is because somebody thought that that flies that have this duplication or deletion have this weird-looking eye, and they thought it looked like a bar. So a normal fly has two copies of the BAR region, normal female, and you can get females that have extra copies, so a duplication in example B, two duplications in example C, or triplication in example D. So no matter how many copies you have, more copies of BAR makes the eye more narrow. Okay, so normal would be two copies of BAR, one on each homolog, but additional copies of BAR make the eye more and more narrow. So that's sometimes called a tandem duplication. We use the name tandem, meaning one right after another, so the duplications are one right after another on the chromosome. So that's an example of a duplication, and it causes a phenotype immediately in this fly. Now this fly, assuming she can see well enough to mate, will be fertile, but it'll be, there will be a compensation loop that kind of allows this to be passed on to her offspring. Now the way we explain how maybe these duplications and deletions first arise originally is potentially through something called unequal crossing over. If you have just a mistake during meiosis, you can have a case where the chromosomes don't synapse properly, and that can cause one of the homologs to get two copies of a gene, and maybe the other homolog doesn't get any of that gene. So that's a deletion and a duplication simultaneously. And then these chromosomes will be passed to the gametes. So you have an individual who gets two copies of BAR from the mother's chromosome and one copy of BAR from the father, then you immediately have a fly that has a narrow eye. And then once the fly has unusual copies of the BAR region, you'll tend to see an increase in the unequal crossing over from then on. So it's kind of interesting. It sort of propagates itself. But the point here is that having too many copies of a gene region causes a phenotype. So sometimes you would think, oh, well, why isn't it just, 2 copies of a gene is good, why isn't 3 copies better? But it doesn't work that way. The genes and the gene products in the cell have to interact with other gene products. So it's like a recipe. When you make a cake, you put in, a certain amount of sugar, a certain amount of butter, a certain amount of flour. If you put twice as much flour in, but you only put the normal amount of sugar and butter, you're not going to get a better cake. You're going to get a cake that's all out of balance. It's not going to be good. So having too much of a gene is not good. It puts things out of balance. I think most students understand why missing a copy of a gene would be bad, but sometimes they don't understand why too many copies is not better. But either way, having a missing gene copy or an extra gene copy throws things off. And another example, there's a gene in fruit flies, again, that if it's deleted, it causes a phenotype called notch, so they call the gene notch. So the deletion of the notch gene causes a phenotype. And so you have this, what they called the notched wing. So it is true that in some genes having an extra copy might not be harmful, but like I said, for some genes it can upset the carefully regulated interaction. So here they show a duplication of gene B, why that could be bad. If gene A, B, and C are supposed to interact in equal ratios, then a cell that makes too much B will have an upset ratio. So it's very much like a recipe. Everything is supposed to be in a certain balance. That's the normal recipe. And if things get upset, if you get too much or too little of any of the ingredients, you can have some kind of malfunction. Now, in evolution, gene duplications have been actually pretty important. Sometimes when they occur, and if they don't cause too much harm, what has happened over evolutionary time is that some genes have been duplicated many times to the point that you get within a genome, within a species, within the genome of a species, you'll have many, many copies of the same gene that have arisen over time through these duplications. And like I said, if it's not harmful, then it can be passed on and passed on. And what we think has happened is that as genes are duplicated, sometimes they get mutated a little bit after they get duplicated, and you end up with these groups of genes that have very similar sequences, but because of small mutations, they serve different functions in the cell. So one good example is the globin gene family. There's, maybe 20 globin genes that exist in groups, in tandem groups on a few specific chromosomes in human. And when you compare the sequence of these genes, one after another, they're very, very similar, and yet we know that some globin genes are only expressed in a fetus, some globin genes are only expressed in certain cell types, and so each gene has now taken on sort of a specific role. The Hox gene family, if you've heard of the Hox gene family, that's important for setting up a segmented body structure. That's another example of a gene family. So a gene family is a group of genes who probably originally came from multiple gene duplications, but now functions together to serve different but similar but not identical functions within the organism. Okay, so deletions and duplications we've covered. Now we're going to talk about large chromosome rearrangements that involve really the movement of sections of chromosomes around. So an inversion is where you have a piece of a chromosome that breaks in 2 places. So imagine a chromosome and there's 2 breaks, and the piece in between the 2 breaks flips around and reattaches. So it's like, let me see if I have a picture here. So here's a normal chromosome. Let's say you had a breakage between C and D and another breakage between F and G. If those 2 points broke and this section, D, E, F, flipped over, then it could reattach and you could get this lower picture. That's an inversion. A translocation, which is over here, we'll talk about that in a minute, is where two non-homologous chromosomes exchange sections. It can be an even exchange, it can be an uneven exchange, it can even be where one chromosome has a piece break off and simply sticks to the other non-homologous chromosome, and it's just a one-directional movement. So there's all kinds of ways, but a translocation involves two non-homologous chromosomes that rearrange. An inversion only happens within one chromosome. All right, so there's two categories of inversion. One's called paracentric and the other's called pericentric. These are just classifications. A paracentric inversion occurs when the two breakpoints are on the same chromosomal arm, like two breakpoints on the short arm or two breakpoints on the long arm, so that the piece that flips around and reattaches doesn't involve the centromere, does not involve the centromere. But a pericentric inversion is where you have one breakpoint on the long arm, one breakpoint on the short arm, and so when the piece flips over and reattaches, it does include the centromere. So not a huge difference, just a classification consideration. But anyway, most of the time when someone has an inversion, they're only going to have it on one chromosome, like one copy of chromosome 3. And then their other copy of chromosome 3 is just normal. So we call them inversion heterozygotes because they have a normal copy of the chromosome and an inverted copy. And so that's the most common. It'd be really rare to find somebody who had the same inversion on both copies of the homologous pair. But anyway, so usually you have an inversion heterozygote. And the inversion itself, you're not losing any genes, you're not gaining any genes, so it doesn't directly affect that individual. You won't see like a bar eye or a notched wing. You won't see any physical symptom in that individual because it doesn't really affect that cell directly. But what it does do is it causes problems in meiosis. And so even though that individual is normal, they have a fertility problem. They can't make gametes normally. So let's look at this. So I said the main problem with inversions and translocations is that they usually don't disrupt or affect genes. It's just that they cause problems with meiosis. All right? So the problem is that the inverted chromosome or the translocated chromosomes do not synapse well. So here's an example of, let's say you have this chromosome here. It's in the replicated form at the top, A, B, C, D, E, F. That's the normal chromosome. Next to it, you have A, B, E, D, C, F, G. So you have the inverted chromosome. So the only way that these two chromosomes can synapse in prophase 1 would be for something like this to happen down below. I think you can tell it's a mess. Plus, you have a crossover trying to happen down here. And so you end up with a mess in meiosis. So it says a crossover within an inversion loop leads to the formation of chromosomes that either contain two centromeres, we call those dicentric, or no centromeres, we call them acentric. And don't confuse acentric with acrocentric. Those are two different words. Acentric means no centromere. And you will get chromosomes with duplications and deletions, so all kinds of problems. So gametes that get these kinds of chromosomes segregated to them will not be able to form zygotes. So an individual with a heterozygous inversion may not have a visible problem other than having low fertility, reduced fertility because of the meiosis problem. Now, interestingly, inversions have been important in evolution, those that don't cause, major fertility problems, because when we compare species, for example, they picked chimpanzee and human, two closely related species, and they gave a specific example, chromosome 4, although you can find lots of examples with all the other chromosomes. But you can see that what they're trying to show you here, and what's true, is that on human chromosome 4, it's only different from chimpanzee chromosome 4 by a single pericentric inversion, which you can see here, this section here. Other than that, the chromosomes are the same. So during evolution, inversions can separate groups into different species, for example. Now, here's what happens if you have a translocation. That's a kind of like a crossover between non-homologous chromosomes. So here you can see a crossover here. And in part B, it shows how those chromosomes, the blue chromosomes are trying to synapse, the red chromosomes are trying to synapse, but then what ends up happening is everything gets synapsed with everything else and you get this mess. And again, you're not going to get normal segregation of these chromosomes during meiosis. So again, reduced fertility is the result. All right, now we have two more types of unusual chromosome structures. One's called fragile sites. These are not very well understood, but a fragile site is defined as a place on a chromosome that's prone to breakage. So they've been observed to break, like a specific place on a chromosome that is prone to breakage in certain individuals. So over 100 fragile sites have been identified in humans, but the most well-studied one causes a disorder. And so we call it Fragile X syndrome. It turns out that some people have a very long CGG trinucleotide repeat on their X chromosome. And if you inherit that, then your X chromosome gets very narrow. I don't know if you can tell on this picture, but it's very narrow, and it will break. And that is correlated to or results in individuals who have intellectual disabilities. If the chromosome breaks, say, before it gets passed into a gamete, then the embryo, the next individual will have a missing piece and you'll have a problem. But that's a pretty well studied fragile site. There are lots of other fragile sites. We don't really know what causes them. They're not all caused by a long CGG repeat. So we know that, but we don't know why other points on chromosomes are fragile. But anyway, a fragile site is just a site that's prone to breakage. Not completely unrelated to our discussion of Fragile X syndrome is a concept called copy number variations. And again, like fragile sites, we don't really know, all the information about this, but by doing the Genome Project, we've discovered that different individuals have more or less copies of certain little sections of chromosomes. And what's different about this is, like with some of the deletions and duplications we mentioned before, you would be able to see some of those. They'd be really large and you would see them on a karyotype. You could tell that a chromosome's shorter or longer. But what if the deletions or duplications exist but they're really small? So we call them submicroscopic deletions and duplications. And like the Fragile X story, that's a case where one individual or some individuals have an unusually long CGG repeat. So that would be that would fit the definition of copy number variation. So sometimes it certainly happens in repetitive DNA sequences. It happens near genes. There are multiple copies of actual genes that have been found in certain individuals who have cancer. But that doesn't mean these things are limited just to these disorders. We don't know exactly sort of the extent of what copy number variation really means or how much of it exists. So we're just starting to scratch the surface on this concept. So I would just think about just the definition of what a copy number variation would be.